linkage proposed by the T.H.Morgan after conducting his experiments on the Drosophila. Morgan and his student C.P. Bridges also conducted the experiments on the same Drosophila in different traits and finally proposed this chromosomal theory of linkage. What is the chromosomal theory of linkage? What it denotes actually? This chromosomal theory of linkage can give answer for this question. That is, how do genes located on the parental chromosomes can be inherited to the next generation? Actually, this linkage is giving one answer for this question. There are other two answers are also there to this question. That is one independent assortment, another one is coupling and repulsion hypothesis. I have discussed about this independent assortment and coupling repulsion hypothesis in separate videos. Those links are given in the description box. You can go and watch them for complete understanding. But here in this session, I am concentrating on this linkage. The parental organisms are maybe homozygous dominant or homozygous recessive or heterozygous dominant or other heterozygotic conditions. But how do the genes or alleles located on these chromosomes can be inherited to the next generation is question here. According to this linkage or chromosomal theory of linkage, there are some linked genes, there are some unlinked genes. Linked genes present on the chromosome can be inherited to the next generation and form the parental progenies. Unlinked genes present on the different chromosomes, they involve in the crossing over, form the new genotypes in the next generation. They can form the new genotypes in the next generation. Briefly, I am comparing this independent assortment, coupling repulsion hypothesis and linkage. In case of independent assortment, the genes or alleles located on the parental chromosomes are separated during the meiotic division and enter into the different gametes and form the new genotypes in equal form. That is independent assortment. But the coupling repulsion hypothesis is different from the independent assortment. If two dominant genes or two recessive genes are present on one chromosome, they couple with each other and enter into the same gamete, they can form the parental progenies. If they are located on different chromosomes, they repel with each other. So they involved in the crossing over, they can form the new genetic recombinations in the next generation. That is coupling and repulsion hypothesis. And now this is linkage. In this linkage, the distance between the genes located on one chromosome and the genetic material present in the chromosome are held responsible for the holding of chromosomes or linking of chromosomes or coupling of chromosomes. In this pattern, the genes present in the parental organism can be inherited to the next generation. That is our main discussion now. In the Morgan's views on linkage, what is linkage? What are linked genes? How do they inherit to the next generation? What are unlinked genes? Why those genes are unlinked? If the genes are unlinked, what type of genotypes are forming in the next generation? What are the causes for the linkage? How do the distance in between the genes is determining the linkage? This is the total discussion. So what is linkage? The inheritance of linked or unlinked genes to the offspring is called the linkage. Means some are linked genes, some are unlinked genes. Inheritance pattern of these genes is discussed in the linkage by the Morgan. Why do genes are linked and why do genes are unlinked? The genes or alleles located on one chromosome are linking with each other because of the genetic material or chromosomal material. The chromosomal material is held responsible to hold the chromosomes. That's why they are linked. They are coupled each other. They enter into the same gamete. Why some of the genes or alleles are unlinked? Because they are located on different chromosomes, and sometimes if those genes are located on same chromosome also, the distance in between the genes is sometimes more. So they were become as the unlinked genes. If those genes are unlinked, there is no attraction force in between them. The genetic material do not involving linking those genes. So what happens? Those are separated, involved in the crossing over, form the new genetic recombinations and new genotypes in the next generation. So from the linked parental progenies from the unlinked new genetic recombinations are produced in the next generation. That is the main discussion here. How it is going on, let us prove. I'll tell you one principle. I'll discuss two examples, then you can understand the complete concept. After completing this entire discussion, you can remember this principle, that's all. If two genes are located very closely on the chromosome, means if the distance is very less in between the alleles, they are linked with each other, hold together, enter into the same gamete. Why they are entering into the same gamete? Because there is no crossing over. So that means if two genes are located very closely on the chromosome, means if the distance is very less, they are linked with each other, holding together and enter into the same gamete without any crossing over. If there is no crossing over, they can form the parental progenies. But if the two genes are located very distantly, if the distance is very wide in between the genes located on one chromosome, they are unlinked. They are unlinked genes and live together. They repel with each other and enter into the different gametes. They involved in the crossing over. They can form the new genetic recombinations. They form new genotypes in the next generation. So the distance is determining the genotype and phenotype of the next generations. The distance is determining whether the genes have to be participated in the crossing over or not. So let us see our example. In this example, these two are the homologous chromosomes. On these homologous chromosomes, these are dominant genes, these are recessive genes. For example, in these dominant genes, in between the AB, the distance is very less. In between the CDE also, the distance is very less. But in between these two genes and these three genes, the distance is wide. This distance is more. As well as the distance in between these genes and these genes also very far. So the distance is more here. The distance is very less in between them. If the distance is very less, the chromosomal material present in between these genes is facilitating to hold the genes firmly, coupled, linked. So they are not dividing. They are not involving in the crossing over. They enter into the gametes as it is. But here the distance is very wide. There is a chance to involve in the crossing over. So if the crossing over takes place, the chromosomal segments are exchanged in between the non-sister chromatids during the packaging stage of meiosis. So these segments are exchanged like this. So this is indicating that if the distance is more, there is a chance to crossing over to form the new genetic recombinations. If the distance is very less, they are linked genes, they do not involve in the crossing over, they do not form the new genetic recombinations, they can form the parental progenies in the next generation. There are actually two kinds of linkages. One is the complete linkage and the other one is the incomplete linkage. This is the example for complete linkage. This is for incomplete linkage. In Drosophila male organism, the complete linkage is found, and for the same genes in the female Drosophila, the incomplete linkage is found. So in the same Drosophila, in males the complete linkage, in the females the incomplete linkage is formed. So here another factor is involving in determining the linkage. That is what we will see. One pair of alleles are dominant alleles and another pair of alleles are recessive on this parental chromosome also. This is B plus means that can determine grey body color, and the VG means vestigial. This is recessive character. Grey is dominant character and vestigial is the recessive character, wing shaped vestigial. So on these chromosomes in the parent, BB indicating the black body color, that is recessive character, VG plus or dominant character which can determine the long wings. So this is black lung. The cross in between these two parents, during the meiotic division only one type of gametes are formed. There is a chance to form only one type of gametes. Whether the crossing over take place or not, they can form only one type of gametes. During the fertilization, the gametes are fused, the F1 generation is formed. The F1 generation is grey long, heterozygous condition, because two dominant genes are located on different chromosomes. Two recessive genes are also located on different chromosomes. So this is heterozygous, but grey long, dominant characters are appearing, recessive characters are suppressed. So from this F1 population, one male Drosophila is taken. This is test crossed with the female organism. So here the question is, why do they crossed this male F1 with the double recessive parent? Means the double recessive parent is a neutral parent. Actually, they can form only one type of gametes. The main aim of the test cross is to know the genetic pattern of the F1 hybrid. The F1 dihybrid is test crossing with the double recessive parent. They were expected that all the progeny produced in the next generation may be in equal quantities. But what type of genotypes they were got in this experiment is, finally they got only two types of phenotypes and genotypes in the next generation. Those are grey vestigial and black lung. Grey vestigial and black lung are the parental progeny actually. When the F1 dihybrid is test crossed with the double recessive parent, they are forming only parental progenies, means there is no crossing over took place in between these chromosomes. So they formed only two types of gametes, parental type gametes. These two gametes formed by this F1 parent are similar like the gametes produced by the parental. These are same. This gamete is also same like this. So with these gametes, they can form only parental type organisms. Grey vestigial and black lung organisms, those are parental types or parental progenies. Grey vestigial, black lung. What is happened here is, in the F1 parent, there was no crossing over. If there is no crossing over, means the genes located on this chromosome are coupled with each other or linked with each other. On this chromosome also, the genes are coupled or linked with each other. They do not involve in the crossing over. They enter into the same gamete. If the parental organisms are formed, that is complete linkage. In the next example, from that same F1 population, this time the female organisms were chosen for the test cross with the double recessive parent. But this time they got 4 genotypes or 4 phenotypes. Grey vestigial, grey long, black vestigial, black long. Means these two are parental types, these two are new genotypes, genetic recombinations. How do these two genetic recombinations are formed? They are formed from these two gametes. How did these two gametes are formed? These two gametes are formed from the crossing over. Why do the crossing over is took place here? Because they were unlinked genes. They involved in the crossing over. That's why new genetic recombinations are produced. They formed a new genotypes in the next generation. Of course, 83% of organisms are parental types, but 17 to 80% of the individuals in this population are recombinants. These recombinants are formed from the unlinked genes. That's why they involved in the crossing over, form the new genetic recombinations in the next generation. So this is incomplete linkage. Means from these two examples, concluded that in the male organism, the body color and the wing shape determining genes are linked genes, but whereas in the female, those genes are unlinked genes. I would like to raise some questions here. Why do the genes in experiment 2 involved in the crossing over and entered into the different gametes? In experiment 2, why do the genes are involved in the crossing over? The genes are alleles located on the chromosomes are involved in the crossing over because they are unlinked genes, so involved in the crossing over. form the new genetic recombinations, new genotypes are formed in the next generation. That is the experiment 2. The question is continued. Why do genes in the experiment 1 were not involved in the crossing over and entered into the same gametes? Because the genes are linked genes. That's why they were not involved in the crossing over. So no new genetic recombinations are formed. They formed only parental progenies in the next generation. So the answer is, in the experiment 1, the paired genes for body color and the wing shape are completely linked, but whereas in the experiment 2, in the female organism or female Drosophila, the genes were incompletely linked. So they were formed the new genetic recombinations. Another interesting question is, total offsprings in the experiment 1 are parental types, 100%, 50:50 ratio. So majority of the offsprings in experiment 2 also parental types, 83%. Why? Means either in the experiment 1 or in the experiment 2, most of the organisms are parental types. Why? Means in Drosophila, the genes responsible for the body color and the wing shape are actually linked genes. In the female Drosophila, these linked genes are somewhat incomplete. That's why they formed only 83%. Remaining 17% are genetically recombined. New genotypes are formed. But whereas in the male Drosophila, these genes are completely linked, so they were formed 100% parental progeny. Now I am going to analyze and discuss some of the questions which is derived from previous competitive examinations. Then the question is, linked genes are located on different chromosomes of the same size and shape, located on the same chromosome, rarely inherited together. So linked genes means they are located on one chromosome, they hold by the genetic material, so they are located on same chromosome. Another question is, linkage is dash as the distance between two genes is dash. What is happening here and what is happening here? Means linkage is dash happened when the distance in between the two genes is dash. These are the two genes located on one chromosome, so they are linked genes. How much linkage force is there is determined by the distance in between these two genes. If the distance is very less, the linkage is more. If the distance is very far, the linkage will become as loose. They will become unlinked genes. So if the linkage is increased while the distance is decreased, so the linkage is inversely proportional to distance. Means one is increased while the another one is decreased. Means this is the principle. So the linkage is, for example, if this answer is put in these two blanks, linkage is decreased as the distance is decreased. This is not correct. Unaffected decreased. This is also not suitable answer. If the linkage is decreased as the distance between two genes are increased. Yes, this is the right answer. And what about this fourth one is, linkage is increased while the distance is increased. This is also wrong answer. That linkage and distance are not directly proportional. They are indirectly proportional. With this principle, we can get the answer. So the answer is C. Means if the distance is more, they tend to be involved in the crossing over. If the distance is decreased, the linking capacity is increased. If the distance is increased, the linking capacity is decreased. So the linkage is indirectly proportional to the distance. The result of linkage. What is the result of linkage? That means complete linkage. What is the result of complete linkage? Complete linkage means The genes are alleles located on one chromosome are completely linked. If they are completely linked, there is no matter. They do not involve in the crossing over, they do not form the new genetic recombinations. We do not expect the new genotypes in the next generation. They can form only parental organisms. In this example already we discussed that. In the male Drosophila, these genes are linked, so they formed only parental progeny. That is the direct answer. You can search for this more phenotype. This is not formation of more wild phenotypes. No answer. Formation of more parental phenotypes. Right. This is answer. Formation of more recombinant. More recombinant are not formed. If two genes are unlinked, the recombination frequency will be. If two unlinked genes are present on the chromosomes, these are the two chromosomes. On these two chromosomes, these two genes located on the chromosomes are unlinked genes. These unlinked genes, how much percentage they have to involve in the crossing over and how much percentage they have to not involve in the crossing over. Means from these two chromosomes, they formed 4 gametes. In these 4 gametes, 2 are the parental gametes and 2 are recombinant gametes. Means 50% are parental gametes and 50% are recombinant gametes are formed. So there is a chance of recombination is 50%. Coupling and repulsion are 2 phases of. Actually, in coupling and repulsion hypothesis, Bateson and Punnett said that coupling means the genes located on one chromosome are coupled with each other. The Morgan said that the genes located on one chromosome are linked with each other. So these two are same, means coupling is equal to linkage. So the coupling is one side of the coin and the linkage is another side of the coin. So coupling and repulsion are the 2 phases of linkage. This is another question from the previous examination. In Drosophila, one allele for red eyes is dominant over pink eyes. The pink eyes is recessive allele. Normal wild type grey body color is dominant on black body color. A researcher mated the homozygous dominant with a homozygous recessive and got the F1 dihybrid, males and females, both organisms. This F1 male is crossed with a double recessive parent. This is test cross. Pink eyes, black body color female, and predicted that the researcher predicted that the F2 were being in equal quantities or equal numbers. Equal number progeny has to be formed. But it became wrong. He did not get the equal number of progeny. The F2 result was red grey with 49%, red black were 4.2%, pink grey were 4.2%, and the purple black were 49%. Finally, the researcher concluded that in a homozygous dominant parent, the red eye and grey body color genes were linked. If so, what is your explanation for the red, black and purple grey offsprings in F2? The question may be long, but we have to fragment the question into different parts. Then we can get the answer easily. Drosophila, an allele for red eyes is dominant on pink. Normal wild type grey body color is dominant on black body color. This is double dominant parent, double recessive parent. So we have to draw the chromosomes. 2 parents are taken. On these 2 parents, we have to label the genes. Red eyes and pink eyes. Red eye color is denoted by the capital letter and pink eye color is denoted by the small letter. Then another character is grey body color is dominant. This is denoted by another capital letter or any other. This is dominant or labeled. These are recessive or labeled. These are black color. The researcher mated the homozygous dominant with the homozygous recessive. This is homozygous dominant. This is homozygous recessive. These 2 are crossed and the researcher got F1 generation heterozygous or dihybrid. F1 dihybrid males and females. So in this heterozygous, some are male and some are female. this F1 male crossed with the double recessive parent. F1 male is taken, means this is F1 male crossed with the double recessive parent. This is the test cross. The researcher want to know the genotypic, the researcher want to know the genes located on the F1 dihybrid. The researcher predicted that all the F2 were being in equal numbers. 4 types of gametes are produced. From these 4 types of gametes, he predicted that all the progeny will be in equal ratios, 1:1:1:1:1. This is independent assortment, but the researcher did not get this. In equal numbers, but it was became wrong. He did not get the equal ratios. Instead of that, he got in the F2 results red gray, red black, pink gray and purple black are formed in this manner. 48% are these, 3%, 3% and 46%. From this ratio, he concluded that the genes located on the same chromosome are linked with each other. So, if they are linked with each other, most of the gametes produced from this parent are these and these. Very less number are involved in the crossing over and formed these two recombinant gametes. So, they formed majority are parental types and few genotypes are recombinants. If so, what is your explanation for the red black and purple gray offsprings? Okay, however, majority of the organisms are parental organisms, but what is your opinion to form these genetic recombinants? Because the alleles are completely linked. If they are completely linked, only all the organisms have to be parental types. So these are not completely linked. So this answer is wrong. They are formed from independent assortment. He did not get 1:1:1:1 ratio. So this is not the independent assortment. They are formed from no crossing over. If there is no crossing over, no new genetic recombinations. This is also wrong answer. So the alleles are incompletely linked. So this is the right answer, because these alleles are linked but incompletely linked. That's why they involved in the crossing over. Few number of genetic recombinants are also produced. That's why they formed new genotypes in the next generation. This is the answer.